Hi there, I hope everyone is staying safe and healthy right now. And I wanted to give you a quick glimpse into the life of a scientist now that everything has been going on and labs are closed. So my name is Cordova. I'm a 5th-year Ph.D. student at the University of Texas at Austin in chemical engineering. And for all of those of us who are not working on coronavirus-related treatments or vaccines, all of our research labs are closed. And so we're all working at home just like everyone else. And so this has given us a really great opportunity to catch up on some reading and some writing. And specifically, I've gotten the opportunity to write up a paper to release some of our work to the public and tell everyone what we've been up to. And so more generally, my work involves engineering yeast to produce useful chemicals and compounds. Just like all living things, yeast take in nutrients or food and release waste products. In typical yeast fermentations that you might think about in making different types of brewing or kombucha, or even in bread, the yeast will eat sugars and then release alcohol and carbon dioxide. However, in my work, we use a different type of yeast called Uroea lipolytica, and we use this yeast because it can make a lot of fats. So instead of that alcohol, it makes fats instead, and we can originally use those fats as fuels, but my work has been actually trying to expand what our cells can make and also what they can eat. And so the first part of my work was expanding those output products. And so the first thing that I've been working on was engineering for higher production of omega-3 fatty acids. And so these are very useful molecules, specifically alpha-linolenic acid, is present in a lot of different foods, specifically eggs and peanut butter, that are added as a supplement due to their health benefits related to cardiovascular health. In my work, we made very precise genetic changes. So we introduced a single group of DNA, and that enables the cells to now make this molecule from the native fats that they already have. And so some of my work was able to generate up to nearly 30% of alpha-linolenic acid of all of the fats in the cell. And then in a similar strategy with a slightly different product, we've engineered our cells to also make a class of compounds known as fatty alcohols. These are used widely in cosmetics, as well as face washes and shampoos. And so now we can source both of these types of molecules that are useful in different applications from yeast rather than from oil or other less renewable sources. And so, as I mentioned, yeast need to eat as well. And so more recently, my work has shifted to changing what the cells are eating. So right now, if we have them eat sugar, that's also a food that the humans can eat. And so if instead we can get them to eat waste, say from something like oil processing, then that can remove the waste as well as generate additional value. And so my research has been using a waste product that's rich in organic acids, so in short, small molecules, and we're using that to feed the yeast, so it's now no longer competing with the human food supply. And so I've actually been spending a lot of time at home writing up this project and getting ready to tell the world about all of the cool stuff that we've been able to make and also how we can detoxify this waste. And this paper is a major part of the scientific discovery method in terms of we need to share what we're doing, that it doesn't matter if we're doing great work if we don't tell everyone about it. And so after the thousands of years that humanity has been working with yeast, starting from bread and beverages, to a variety of other products that we see now, scientists using my work can generate even more useful products that we need every day in our daily lives, and sometimes we can even get that from our own waste.